Okay, Samuel, so we've built this lung tumor classifier. Seems pretty solid, right?
Yeah, pretty good. But we're hitting a snag.
A snag? What's the problem?
We're relying on hand-labeled nodule candidates. That's super time-consuming.
So, manually labeling thousands of images? That's a big bottleneck.
Exactly. And the sheer volume of data. It's crazy. Imagine slicing up every CT into tiny cubes. 31x31x7.
Whoa, 6,727 patches per CT. That's massive.
Yeah. Way more data than we have labeled examples. Positioning matters too. Our classifier needs the nodule right in the center of the patch. Otherwise, it might not work properly.
So, what's the solution? We can't just feed the whole scan?
Right. We need a way to automatically identify and pinpoint those nodules.
You mean like a pre-processing step?
Basically, a way to find the nodules before feeding them to the classifier. It's a multi-step approach. We have different steps.
That makes sense. In real-world medical applications, this multi-stage approach seems common. Why do researchers sometimes try to cram everything into one deep learning model, which isn't always practical?
Well, in academic papers, researchers want to prove their models can handle complex tasks. That's a good goal, but in real clinical settings, efficiency and interpretability matter more.
So, we're looking at building this stage by stage. This is a good strategy to make each stage testable.
Absolutely. It allows us to build the system in more manageable modules.
That sounds like a better route.
And makes everything more understandable, too.
All right, Samuel, so we're diving into segmentation now. This is step 2 in our lung cancer detection pipeline. What exactly is segmentation, and why is it crucial here?
Segmentation, in simple terms, is like precisely identifying and marking areas of interest within an image. In our case, we're looking for potential nodules in CT scans. It's crucial because without knowing where the nodules are, our classifier can't effectively assess if they're cancerous or not. It's like trying to identify a specific item in a cluttered warehouse. You need to know where it is first.
Got it. So, we're using a UNet model. Tell me more about that.
UNet is a type of neural network architecture specifically designed for image segmentation tasks. It's known for its ability to handle spatial information effectively. Think of it like having multiple layers of filters, each looking at different parts of the image, working in tandem to pinpoint those nodules.
Okay, so we're essentially training a model to draw on the images, right?
Precisely. Instead of simply saying yes or no, this model outputs a whole image showing where it believes nodules are located. It creates a mask highlighting the suspected nodule areas. It's a more granular approach compared to our previous classification step.
How does the dataset need to change for this segmentation task?
We need more information. Before, our dataset just focused on small areas around suspected nodules. Now we need full CT slices, plus 2D crops from those slices, for training the UNet. This gives the model a comprehensive view and more accurate location data.
So it's learning from the full context of the image, not just little snippets?
Exactly. It's learning the patterns and features of nodules within the larger context of the whole CT scan. You'd need to use a larger set of labeled data for effective training.
And what about the training loop? What adjustments are necessary?
The training loop needs an updated loss function. Our previous one was suited to binary classification. This time, we need a loss function designed specifically for pixel-level labeling, something that's optimized for the segmentation task. We also need to include methods for saving model weights for later use and visualizing the results in TensorBoard.
Makes sense. So, a much more complex process to train the model, because we're not just looking at a simple binary outcome?
Yeah. We're moving from a simple yes/no classification to a precise delineation of nodule locations. This step demands more data, a tailored loss function, and an updated training loop. All part of building a more accurate detection system.
So, Samuel, semantic segmentation. That's classifying pixels?
Exactly. Like labeling individual pixels in an image. Think of it like painting a picture.
Painting a picture pixel by pixel? That's granular.
Very. You're assigning a meaning to each tiny part. Like marking this is a bear, this is a cat. Really detailed.
And how does this relate to finding nodules?
We're using it to find potential nodule areas. It creates a mask.
A mask? Like a stencil?
Precisely. A binary mask. True for nodule candidates, false for healthy tissue.
So, it's a yes/no for each pixel?
Right. It helps us pinpoint possible nodules, which we can then analyze further.
Interesting. What about other segmentation methods?
Well, instance segmentation labels individual objects. Imagine two people shaking hands. Semantic would label person and background. Instance would label person 1, person 2, and background. It's more precise.
But that's a bit more complex.
Yes. And in our case, the nodules are likely separate, not overlapping. So, simpler grouping works better.
So, why not just use instance segmentation for nodules?
Instance segmentation is great, but for our case, it's overkill and it takes more resources. It's more work for something that is simple enough.
Object detection is another option?
Yes, object detection locates the nodule and draws a bounding box around it. It's useful, but more complex than semantic segmentation. Also, it's resource-intensive. We opted for semantic because it is simple, clear, and doesn't use too much power.
Makes sense. So, semantic segmentation fits our needs better than the others?
For this project, definitely. It's less complex and perfectly suits our task. We're focused on efficiency and simplicity now.
So, Samuel, we're talking about semantic segmentation, right? It's different from just classifying if something's present.
Exactly. It's like finding where something is, pixel by pixel.
Okay, so if I have a picture and I want to know exactly where the cat is, not just is there a cat. That's segmentation, right?
Yep. You need a different model. Classification just tells you yes or no, but segmentation gives you a map, a mask, showing you the cat's location.
So, how does this work? Why does it need a different model?
Well, imagine trying to take a giant image and boil it down to one output. You lose a lot of detail.
Like a magnifying glass. I get it.
Exactly. Classification models act like magnifying glasses focusing on a single point, ignoring pixel-level details. Segmentation needs to keep those details to pinpoint the object's location.
And how does that impact the architecture?
You need a different architecture. Classification relies heavily on downsampling. Each layer shrinks the image, increasing the effective reach. Segmentation needs output the same size as the input, but without downsampling, the reach is too local. We need a balance.
So, upsampling is the solution?
One common approach is upsampling. It's like enlarging the picture, but we need to make sure we do it in a way that maintains the details. We can't just copy the values and enlarge the pixels. You have techniques like linear interpolation, or using a model to learn how to upscale the image to get the details back in.
I see. Upsampling is a key part of balancing output size with sufficient receptive field?
Exactly. Without it, each segmented pixel could only consider a tiny neighborhood. Upsampling helps in this sense by building back the important context. We need layers that look at a larger and larger area to identify details. Downsampling helps for classification tasks, but hurts for segmentation.
So, what kind of architectures could be helpful for upsampling?
We could have repeated convolutional layers, without downsampling, but that limits the receptive field, making it hard to capture distant details. It needs a strategy to make the model's effective reach larger.
It seems crucial to balance the receptive field size with output size. It's a trade-off, right?
Absolutely. Adding max pooling between convolutional layers expands the receptive field while downsampling the image, but to get the needed pixel-level output, we need to strategically upsample to maintain the details lost from downsampling.
So we can also adjust receptive field sizes by strategically inserting max pools, right?
Precisely. You can adjust the receptive field size by controlling how many convolutional layers are stacked, and by strategically inserting max pooling. It's about finding the right balance.
That sounds like a challenging balancing act.
It is. But upsampling gives us a crucial tool to solve that issue.
Okay, Shamel, UNIT. This sounds pretty crucial. Can you give us the gist of it?
UNet's a segmentation network, designed to give pixel-by-pixel outputs. Think of it like an incredibly precise painter, not just identifying the object, but highlighting each pixel of the object.
So, it's more than just classification?
Precisely. It's fundamentally different from classifying things like images as dog or cat. UNet focuses on precisely pinpointing each pixel belonging to a specific object.
And the U-shape?
It's the architecture. It's a convolutional neural network, with a series of downsampling and upsampling layers. Imagine a funnel, shrinking the image data, analyzing it, and then expanding the data to rebuild the original resolution.
Downsampling and upsampling? Explain that.
Downsampling is like looking at a zoomed-out image, reducing the resolution to focus on larger features. Upsampling is the reverse, rebuilding the resolution. Both steps are crucial for accurate segmentation.
How are skip connections important here?
Skip connections are vital for maintaining spatial information. As you zoom out, losing details can lead to inaccurate reconstruction. Skip connections reintroduce critical data to later stages, ensuring the network captures fine detail. It's almost like having hidden shortcuts from one part of the process to another.
It's almost like a shortcut to a better picture. That's brilliant. And the final one, by one convolution?
That's the classification step. It takes the output, reduces the number of channels, finally matching them to the needed output classes. It's almost like narrowing down your focus to the exact types of objects you want to identify.
So, this UNet is a powerful tool for segmentation tasks. Tell me more about the receptive field concept.
Yeah, receptive field is important. Early convolutional network designs struggled with limited receptive fields, which meant they couldn't see the big picture as well. UNet's design fixes that by going from small detail to the wider scope, and back again, like zooming in and out to see the overall object context.
Wow, that is a lot to unpack. So, UNet addresses issues like spatial information loss during downsampling?
Exactly. It solves issues in earlier models by maintaining more information through the skip connections, keeping accurate object boundaries during downscaling and reconstruction.
I see. This is why it's a breakthrough. It's quite different from other methods.
That's right. Its design allows it to learn from both a fine detailed view and the wider picture simultaneously.
Okay, Samuel, so we're moving to segmentation. This is a pretty big step up.
Yeah, it's moving from just saying yes or no to a pixel-by-pixel probability map.
So, instead of a simple binary output, we're getting a probability for every single pixel. That's a huge leap. How does this work?
Exactly. We're using a pre-built UNet. It's a type of neural network designed for image segmentation.
So, instead of building this from scratch, why not build it from scratch?
Well, it's way faster this way. A pre-built model has been tested and optimized, and we can adapt it to our needs rather than starting from scratch. It's also about building a toolbox, a portfolio of different tools that we can use.
That makes sense. And this UNet from GitHub, is that like a standard model for this kind of thing?
It seems to be a good choice for our use case. We gotta make sure it fits our data though. And remember to check the license.
Absolutely. So, what are the key components that we should look for when we're looking at this UNet code?
We need to understand its architecture. Skip connections, for instance, are really important because they let the model reuse useful information from earlier stages to avoid data loss.
Okay, so skip connections are a good way to avoid losing information. That's smart. Can you explain why this is important for image segmentation in general?
Well, imagine you're trying to identify different cells in a microscope image. The earlier layers might recognize basic shapes, and skip connections link those details to the next layers to refine and identify the actual cells.
Got it. So, we're essentially building upon the layers of processing by reusing some important information from earlier stages?
Precisely. Plus, it helps with training efficiency.
Excellent. So, we're picking a pre-built model, examining its structure, and tweaking it to fit our specific image segmentation needs. A very smart approach. Okay, Samuel, we're adapting a UNet model. So, batch normalization. Why is that important here?
Well, Alfred, batch normalization helps standardize the input data. It's like prepping ingredients for a recipe. Without it, you could have varying input strengths, and the model might overreact to some inputs.
So, it's a pre-processing step?
Precisely. It also estimates the mean and standard deviation of the data in batches. This is super useful. Imagine a batch with really weak signals. The normalization will scale those samples more intensely.
That makes sense. But why is that a good thing?
Randomly picking batches at each epoch means a dull batch won't consistently affect the model's training. It avoids these outliers skewing the learning process. It's like balancing out the ingredients in cooking.
Interesting. What about the sigmoid layer?
The output values of a U-Net are unconstrained. A sigmoid layer ensures the output is between 0 and 1. It's a probability.
So, it's clamping the output?
Exactly. Now the model's output is more meaningful, like a probability estimate. It makes the results easier to interpret.
Reducing depth and filters. What's the reasoning?
The initial model's capacity might be too high for your dataset. It could overfit, learning noise instead of true patterns. Like using a giant meat cleaver to mince vegetables, you're likely to damage them.
You mean it could be overkill?
Yeah, exactly. It's like having a sports car for short errands. You might not get the best mileage, and it's more complicated than you need. It is a big trade-off between power and effectiveness in deep learning.
So, you're basically simplifying the architecture?
Yes, reducing depth and filters, and choosing an appropriate size for the dataset. By simplifying the model, you get a more efficient and adaptable model.
The 2D approach instead of 3D. Why not fully 3D?
The memory needed for a 3D U-Net would be substantial, and probably not practical given our CT scan data. The spacing in the axial direction is larger, so using adjacent slices is helpful for detecting something like a blood vessel. It's like looking at a picture, then a picture beside it for a more complete understanding.
So, it's a trade-off between computational cost and the utility of the third dimension, right?
Exactly. A simplified 2D approach still gives us the context from adjacent slices.
Okay, Samuel, we're updating the dataset for segmentation. What's the big change here?
We're switching from 3D to 2D input and output.
So, the model's expecting a different format?
Exactly. Our original U-Net produced 3D segmentation maps. Now it needs 2D.
Why the change?
Well, the original model's approach was pixel perfect. Every output pixel had a complete receptive field.
So, no padding?
Right. No extra information. It works well for some applications, but it's got limitations with convolutional downsampling when we're dealing with 3D data.
So it's about how the downsampling and convolution interact?
Yeah, it's a bit tricky. And second, our data is three-dimensional. That's a challenge with this new 2D format.
I see. How does this pixel-perfect approach cause problems when working with 3D data?
The problem is, when downsampling, it can lose essential information from the 3D volume. The 2D slices might not fully represent the spatial relationships within the full volume.
That makes sense. Are there any techniques to address this issue?
Absolutely. We can employ techniques like padding. Imagine adding extra blank areas around the 3D data.
That preserves information?
Yep. By adding padding, we retain more context within each slice. This better reflects the 3D nature of the image. It's like getting a wider view.
So, we're adding buffers around the input data?
Exactly. This helps us keep important information from neighboring slices in our calculations, avoiding losing crucial context. Essentially, it gives us more context for each 2D slice.
So, is padding crucial for the new model to function properly?
Absolutely. Without proper padding, you can lose important details. This isn't just about maintaining precision, it's about the accuracy of our model segmentation.
So, Samuel, U-Net's input size is crucial, right?
Yeah, absolutely. It's a specific requirement.
Why is that? What's the connection between input and output sizes?
U-Net layers have downsampling and upsampling steps. To get accurate results, sizes need to match.
Can you elaborate? Like, what does that mean practically?
Imagine a conveyor belt. Each layer shrinks the image.
And the input-output sizes?
They need to line up. Otherwise, you lose data.
Like a puzzle then?
Exactly. The 572 by 572 input creates a 388 by 388 output.
Okay, But our CT scans are 512 by 512. That's a problem, right?
Exactly. Nodules near the edges won't be fully segmented.
So, what's the solution?
Padding. Adding extra space.
Padding? Like, around the edges?
Yep. Makes input size flexible.
So, we're sacrificing some accuracy near the edges?
Potentially, since padded areas are artificial. But that's the trade-off.
A good trade-off if it works better overall?
It often is. Gain in applicability.
So, padding is a good solution, but not perfect?
Yeah, a worthwhile compromise. Accuracy versus broader coverage.
Helpful context. Makes sense. Okay, Samuel, so we're tackling 3D UNet. You mentioned trade-offs. What's the big problem with feeding a 3D image directly?
Yeah, the raw 3D image size is huge. Imagine a 512 by 512 by 128 image. It's a lot of data.
So, memory issues?
Exactly. The first layer of a UNet often has many channels. With that size, our GPU just can't handle it.
That's a significant hurdle. What's the workaround you described?
We're treating each slice as a 2D segmentation problem. Think of it like stacking images, one after the other.
So, instead of a 3D volume, we're working with 2D slices? How does that impact context?
We're using neighboring slices as separate channels. So, slice above, slice below, and so on.
Interesting. So, a slice above becomes a separate feature?
Precisely. It gives the network some depth context.
But what about the spatial relationships? Are you not losing something critical?
Absolutely. The connection between slices isn't explicitly represented. It's all kind of averaged out. The convolution kernels combine everything linearly, so we don't know if a feature is from one slice or the next.
So, it's a trade-off. We gain some memory, but we lose the full 3D relationship.
Exactly. We lose the wider view from downsampling in the depth dimension as well. It's all about compromise.
And what about slice thickness?
We're ignoring exact slice thickness. The model needs to learn to be robust to different spacings. This needs to be addressed in the data.
So, it's less about the perfect 3D and more about creating a robust model?
Exactly. And there's no magic formula. You know, there's no one-size-fits-all answer. It's trial and error, testing hypotheses systematically.
So you're advising against multiple modifications at the same time, right?
Absolutely. It's too easy to have interactions and not know what's actually working.
So, Samuel, we're building ground truth data for our model. Why this mismatch?
Right. Our human-labeled data gives us points, but we need a mask for every voxel, indicating if it's part of a nodule. We've got to create that mask ourselves.
Makes sense. So, manual checking?
Yeah, we need to validate those handmade rules. It's tough at scale. We're not going to meticulously check every nodule.
A few test samples then?
Exactly. A quick looks reasonable check. We need easy ways to look at intermediate steps in the algorithms, even if the functions are a bit clunky.
Why is that important?
Debugging and understanding what's going on. Visualizing helps a lot.
Got it. Bounding boxes. How do these help?
We convert nodule locations to boxes, covering the entire nodule. We trace outwards from the center. That's key for finding lung tissue, avoiding overinclusion, right?
And how do we find the boundaries?
We look at voxel density. We stop when we hit low-density voxels, air basically. It's a radius expansion in all directions.
So, the code we saw, what's its core purpose?
It implements that search for bounding box boundaries. It loops through, checking density, handling potential tensor boundary problems. The radius stops when density drops below the threshold.
Why the one voxel border?
Some nodules are close to lung walls. We have to check both directions. That ensures the box doesn't include extra lung tissue. It's about accuracy.
So, creating the mask.
We loop through each nodule, find its box, and mark the corresponding voxels in the mask. Then, we intersect this mask with voxels denser than our threshold.
Why intersect?
That clips off extra lung wall voxels, improving the nodule mask accuracy. It fits the nodule better.
Interesting. So, we cache masks too?
Yes. We're caching chunks of CT data around nodules, and now we cache the positive masks for better efficiency in training. This makes training speedier.
And the cleaning of annotation data?
We're fixing issues in our annotation files. Apparently, some nodule annotations are repeated, not just duplicates. We're using a cleaned annotation file from a larger, well-supported dataset for improved quality.
How does this improve our model's accuracy?
By using a reliable dataset, we get better, cleaner nodule data, reducing noise. Using a cleaner source helps our nodule masks be more reliable, hence, a more accurate model.
Okay, Samuel, so this new dataset approach, it's a little different. 2 classes for training and validation? Explain that to me.
Yeah, it's a design choice to improve clarity. Instead of one big dataset, we've got a base class for validation, and a specialized subclass for training. This makes it easier to track which parts of the code only affect training.
So, less code duplication, but more classes? Makes sense. Why not 2 separate datasets?
We considered that, but the approach we chose keeps things a bit simpler, without inventing unnecessary complexity. Standard software practices are key, right? We try not to overcomplicate things.
Makes sense. And these 2D CT slices? They're not just grayscale images, right?
No, they have extra channels. Think of it like an RGB image, but instead of colors, these channels are adjacent slices from the CT scan. We're treating those slices like color channels in a 2D image.
So, this multi-channel 2D image, what about validation? You need a sample per slice?
Exactly. And because the CT scans can have a different number of slices, we're caching their size. We need this to quickly create the validation dataset, without loading the entire CT each time. This cache is populated during a separate pre-processing step.
Ah, the caching decorator. This prepcache.py script. Sounds efficient. How's that caching done?
It's a function, getCTSampleSize, that we decorate with the caching mechanism. It figures out the number of slices and the locations of positive masks in the CT.
And the init__init__ method. What's different?
It takes a context slices_count parameter. Think of it as a window, showing the slice, and a couple slices before and after. Also, we use an augmentation_dict for training, similar to what we've used before. Crucially, we now split the entire CT scan into training and validation sets, rather than splitting nodules.
Gotcha. So, you're treating the whole CT scan as a single unit, rather than individual nodules. That changes everything.
Yes, the validation now supports 2 modes. Full CT_bool equals true for evaluating complete CT scans, and full CT_bool equals false, which uses only slices with positive masks. This is crucial for evaluating true and false positives without bias.
Okay, that makes validation more comprehensive. You're creating a list of series?
Exactly. We loop through the series, get the number of slices, and whether it's a full CT scan or just the positive slices. This is crucial for ensuring all parts of a CT scan are covered and the validation set correctly represents the complete dataset, rather than just nodules.
And the __getitem___ method is modified too, right?
We delegate most of the work to a separate function, getitem_full_slice. This function retrieves the specific CT slice. This is critical for efficiency and code clarity.
So, the main process retrieves an index. The function then calculates the actual sample and retrieves the slice from the cache CT data. Brilliant.
Yes. Everything is pre-processed. This significantly reduces processing time. Plus, we have series UID and slice index information for debugging, while separating these from the training data itself.
Great explanation, Samuel. This more nuanced dataset splitting is crucial for accurate evaluation. It will make a big difference, won't it?
Exactly. It provides a more comprehensive validation framework that accurately measures true and false positives and negatives and prevents bias.
Okay, Samuel. So you're saying we're training on smaller crops? Not whole CT slices?
Yeah, that's the key. We're using 64 by 64 pixel crops.
Why not the whole slice? Seems more data.
Well, it's about training stability. Whole slices, Have too many negative samples.
Negative samples? Explain that.
Imagine finding a needle in a haystack. Nodules are tiny. So the vast majority of the pixels are not nodules. Training on the entire slice overwhelms the model.
So, by training on crops, we reduce the background noise?
Exactly. We're focusing on the nodule and surrounding areas. Taking 96 by 96 crops around each potential nodule, and then random 64 by 64 crops within that.
So, what about validation? That will be different, right?
Correct. Validation uses the whole slice. The same convolutional model, but applied to a larger set. This can make the model look a lot worse for this validation set though.
A lot worse? How so?
Validation has way more pixels that are not nodules. So the model might see many more false positives because there are just more chances to be tricked on validation. We need to take the validation result with a grain of salt.
So more negative samples on validation will inflate the false positives?
That's right, and with our focus on high recall.
High recall?
Yeah, we're trying to identify as many nodules as possible. We're sacrificing some precision for that complete coverage. A major reason behind this strategy.
Interesting. So, this approach is about training speed and stability?
Primarily, stability and quick convergence, because of reducing those negative samples in the training data. We got pretty bad results when training on the full slices.
So, it's a trade-off really?
Absolutely. More stable training, with the cost of some inflated validation metrics. We need to keep that in mind.
Okay, Samuel, so we're diving into the training dataset now. This get item __ method. What's the key difference here?
Well, Alfred, it's essentially the same as the validation sets, but with a crucial tweak. Instead of just selecting a slice, we're pulling specific candidate information.
Right. So, we need more detail. Why the specific center location?
Precisely. For training, we need the whole series and the exact coordinates of the center of the region we're segmenting. It's vital for accurate training.
Got it. And get item _ training crop handles that cropping?
Exactly. It gets the relevant data and then creates those 64 by 64 random crops from the 96 by 96 input.
That makes sense. And the get C to raw candidate function. Is this similar in concept to what we use for classification?
Exactly. It's similar, but it also now returns a crop of the positive mask alongside the CT data. Crucial for segmentation.
So, we're randomly selecting crops, right? How does this avoid potential issues like overfitting to specific data samples?
You're right, random sampling is key to preventing overfitting to specific data points. It's like showing the model many diverse examples, ensuring it learns the general patterns, not just the noise within a single sample.
So we're not just grabbing a slice. We're selecting a region within the slice.
Yes, that's right. We're taking crops around the center location of the slice, rather than simply a slice of the whole image. This allows us to focus on the critical parts that lead to accurate segmentation.
And the random row and column offsets? Are we using standard image augmentation techniques here?
Well, we're augmenting the data in a way that's not explicitly done through a standard augmentation library. This approach is different from normal, more standard methods. We're handling the augmentation work on the GPU for efficiency.
Interesting. Why the GPU? Is it computationally expensive to do this?
It can be. It's about leveraging GPU power for faster processing. Doing this on the GPU means all the transformations are done in parallel. That can really help with speed.
Excellent point. So, this approach trades some traditional augmentation libraries, but targets that crucial speed increase on the GPU?
Exactly. We're focusing on speed and efficiency. It's a trade-off, but a sensible one here.
Okay, Samuel, so we're talking about moving data augmentation to the GPU. Why is that a good idea?
Well, Alfred, GPUs are super fast for certain types of calculations. If the data gets bigger during processing, keeping it on the GPU means less data has to be moved back and forth between the CPU and GPU, which is way faster than the CPU.
Right? Less data transfer means less of a bottleneck. So, we're not just talking speed here, we're talking efficiency, right?
Exactly. Imagine a factory line. You want the bottleneck to be the most expensive or hardest part to improve. If your GPU is the expensive machine, you don't want the data loading or pre-processing to hold it back. So we move the data augmentation to the GPU.
And how do you actually do that in code?
We create a special model. It's just like the other models we've discussed, but this one doesn't calculate gradients. It takes input tensors and produces new tensors. It does the data augmentation. The crucial part is that forward method.
So, a separate model for augmentation. Interesting. Can you break down the forward method a bit?
Sure. It takes input and a label, builds a transformation matrix. Then, it applies this transformation to the input and label using functions like affine_grid and grid_sample. These functions are essentially like applying a 2D transform like rotation, flipping, and offsetting to the image data.
So you're using these transformations to augment the image data on the GPU. How do you define these transformations?
We define transformations within a function called _build 2D transform matrix. It makes a 3x3 transformation matrix. This matrix can be used to rotate and flip the image. The random transformations are done with Python's random library for an element of randomness during training.
Random rotations? That makes it useful for real-world datasets, huh?
Absolutely. It helps your model generalize better to real-world data. If the data has variations, this random augmentation helps the model learn those variations. Without it, the model might overfit to specific patterns in the training data.
So, the data is modified on the GPU and then fed back into the network.
Precisely. And to make it clear, you'll notice it converts the masks to floats, because grid_sample works with float data. Then we put some noise on the input. We add some simulated noise, which is good practice.
You're saying noise augmentation is a thing?
Yes, sometimes, adding noise during the augmentation stage can make your model more robust to variations in the data. It can improve performance on real data where there might be some degree of noise or distortions.
That's really cool, Samuel. So, by moving the augmentation, we're essentially lightening the CPU load and having the GPU do the heavy lifting.
Yes, exactly. It's all about optimizing resource usage and using the right tool for the job.
So, Samuel, we're updating the training script for segmentation. What's the big picture here?
Yeah, we're taking our model and data and training it to better segment things. Crucially, we're adding a dice loss function.
Dice loss? That sounds interesting. Can you explain?
It's a specialized loss function designed specifically for segmentation tasks. It focuses on the overlap between the predicted segmentation and the actual segmentation. This is way more effective than just a general loss function for these kinds of problems.
Makes sense, really highlights the regions of interest. And why Atom instead of SGD?
SGD, well, it's a workhorse, but Atom often converges faster and more reliably, especially with larger datasets. It's a pretty common choice for these situations.
Okay, so Atom Optimizer. Got it. What about logging to TensorBoard?
Right, we're adding image logs for visual checks of our segmentations, and also more metric logs for a comprehensive analysis, using TensorBoard. It helps us track progress and spot potential issues.
So, you're logging images and metrics. And how is the model saved?
We're saving the best model based on validation performance. We want a model that does well on data it hasn't seen yet.
And how does this script compare to the classification one from Chapter 12?
Pretty similar, actually. The big changes are the new model instantiation, the new loss, and the optimizer. But a lot of the underlying structure remains the same. It's just a very straightforward update.
So, this training script seems more robust, particularly for segmentation tasks. Any potential pitfalls to watch out for?
Potentially, hyperparameter tuning. We could spend a lot of time fiddling with those parameters to get the best segmentation performance.
All right, Samuel, let's talk about this segmentation and augmentation model initialization. So, we're using a UNet wrapper, right?
Yeah, it's a pre-built structure for image segmentation. It's really handy.
7 input channels? That's a lot. Can you break that down?
3 regular input channels, plus 3 more that act as contextual information, and then 1 extra channel focused on the nodule we're trying to identify.
So, essentially, we're feeding in a broader picture for better accuracy?
Exactly.
And this depth parameter, what does that control?
It determines how many layers the U-shaped network has. Each downsampling step adds a layer. So, depth 3 means 3 such steps.
What about the WF equals 4 parameter? What's that for?
That controls the number of filters in the first layer. It's 2 to the power of WF, which in this case is 2 to the power of 4 or 16 filters.
So, the layers get wider as you go down and then back up?
Exactly. The network learns progressively more complex features as it processes deeper and then reconstructs.
Padding? Why is that important?
It's essential for preserving the size of the output image. Without padding, the output might be smaller than the input, losing crucial information.
Batch normalization. Why is that in there?
Batch normalization helps stabilize training and accelerates convergence.
So, the augmentation model, what's that for?
This is separate from the segmentation model. It introduces variability in the data to make the model more robust. It's like giving the network many different examples of the nodule.
So, these two models work together, one for learning the features, and the other for making the training data more diverse?
Exactly, providing a strong combination for better, more generalizable segmentation.
So, Samuel, you mentioned Atom as a great starting point for optimization. Why is it so popular?
Yeah, Atom's super common. It's an alternative to the more traditional SGD, you know?
Right, stochastic gradient descent. What makes Atom different?
Atom's smart. It adjusts the learning rate for each parameter individually. SGD uses one learning rate for everything.
So, it's like fine-tuning for each weight in the network?
Exactly. And it does that automatically, as training goes on. It learns a good pace as it goes.
Cool. So, no more fiddling with learning rates manually?
Usually, you don't need to. Atom finds a good one itself, saving a lot of time and experimentation.
Interesting. But what about all those variations? Atomax, Ratum, Ranger? Are they better?
Those are refinements, some with specific strengths, like dealing with certain kinds of problems better. But Atom's usually a good starting place.
So, you're saying there's no one-size-fits-all?
Exactly. There might be cases where a different variation does outperform Atom, but it often depends on your specific network and data. Finding the right one for your exact project can take quite a bit of testing.
Sounds like a huge potential win, but it also means lots of trial and error potentially.
It does, and there can be a lot of testing. It's a balancing act, you know?
So, in practice, would you recommend starting with Atom almost always for a project?
Yeah, almost always. It's a really solid baseline. Unless you have compelling reasons to use something else, it's typically the faster and easier choice.
Okay, Samuel, so dice loss. You mentioned it's useful for segmentation tasks. Can you unpack that a bit?
Sure, Alfred. Dice loss is great when you have uneven datasets. Imagine a CT scan, mostly empty space, a few tiny nodules. Cross-entropy struggles in those scenarios.
Why?
Because cross-entropy emphasizes individual pixels, and the majority class overwhelms the minority. Dice loss, though, looks at the overlap between predicted and actual segments.
So, it's about the overlap rather than individual pixel accuracy?
Exactly. It's like measuring how much of the identified nodule area the algorithm actually captured.
And how does it calculate that overlap?
It's a ratio. 2 times the intersection of the predicted and actual segment, divided by the sum of those areas. Think of it as a sort of precision recall balance.
That sounds familiar. Like an F1 score?
You got it. It's essentially a per-pixel F1 score, but adapted for training. Critically, for training, we need it to be differentiable.
Differentiable? What does that mean?
For an algorithm to learn, we need to know how changing a weight affects the loss. A differentiable function allows us to calculate these changes.
Gotcha. So, this differentiable dice loss, is it soft dice?
Yes, this is soft dice. We're dealing with continuous predictions, not just binary ones. It's crucial for learning.
And you mentioned something about how the output is modified, making it into a loss function?
Yeah, we subtract the dice ratio from 1. This flips the function so that high overlap means low loss, and low overlap, high loss.
Okay, so we're essentially trying to maximize overlap, right?
Absolutely. The code, computes the dice ratio, and then subtracts it from 1 to turn it into the loss function. It's all about getting the positive pixels right and weighing false negatives heavily.
You're talking about different types of dice loss, right?
Yes. In the code, we have a normal dice loss, and then a fine loss focusing only on pixels inside the ground truth areas. Why? Because that fine-tuned loss really emphasizes recall. We want to make sure we don't miss any tumors. So we weight it heavily, often 8 times more than the regular dice loss.
So, that 8 times weighting is to favor recall over precision?
Correct. We sacrifice precision for recall in this specific scenario because it's more critical to find all the nodules than to get perfectly accurate boundaries for the nodules.
And what's the point of the 0.5 threshold?
We're converting continuous predictions to binary yes or no for measuring true positives, false negatives, and false positives. It doesn't matter if the prediction is 0.6 or 0.9, as long as it's above that threshold.
So, we're explicitly favoring false negative avoidance in our loss function.
Precisely. This approach is very useful when using an atom optimizer. SGD might lead to every pixel being classified as positive.
So, atom optimizer helps manage the training process in this case?
Yes. It lets us fine-tune the learning rate, and this prevents the false negative loss from being too overpowering.
Okay, Samuel, so we're talking about visualizing segmentation results in TensorBoard. This is crucial, right?
Absolutely. Visualizing is key to understanding model performance. You want to see if your model is learning correctly, or if it's going astray.
So, we're logging images now, not just metrics?
Exactly. And we're doing it in a way that's efficient and informative.
How do we pick those images? Do we just grab random slices?
No, we want a representative view. We're taking 12 whole CT scans, and from each, we pull out 6 slices evenly spaced throughout the scan.
Why 6 slices? And why 12 scans?
TensorBoard handles 12 images at a time. We space the slices to get a good overview. 6 slices give us a decent spread.
Okay, so we have these images, but how are they presented?
The top row shows the ground truth, what we know to be correct. The bottom row shows what our model predicts. We overlay this in a way that's easy to compare.
How do we know what's right and wrong, though? What are the colors?
We use colors to highlight different kinds of errors. Red for false positives and negatives, orange for false negatives, and green for correct predictions. This lets us easily spot problems in our model segmentation.
Right, so it's like a visual report card.
Exactly. Crucially, we can also see how the model's predictions change over time. We can see the model's progress with a slider in TensorBoard.
That's really useful for debugging. And you mentioned using CT scans. Why CT scans, specifically?
CT scans are commonly used in medical image segmentation tasks. It's a crucial type of medical imaging for visualizing structures inside the body.
Interesting. So, this image logging is part of our training loop? How often do we do this?
We validate and log images on the first epoch, and every 5th epoch after that. It balances validation frequency with the amount of training time we spend on the GPU.
You're trying to maximize training and minimize validation time. That makes sense.
Absolutely. We want those insights quickly, but also want our models to train for a longer period to improve performance.
Are there any common pitfalls or troubleshooting issues when doing this?
One potential problem is that a huge number of images might overwhelm the browser. TensorBoard will limit the display to avoid that, but it's worth monitoring. Also, the code ensures that the CT intensities are in the 0 to 1 range for proper display in the images. Otherwise, you can get weird results and potentially miss important information in the visualization.
So, we're logging metrics now, Samuel. What are these metrics telling us?
We're calculating things like true positives, false negatives, and false positives, standard evaluation metrics for classification tasks.
And why are these important?
Crucial for understanding model performance. We're looking at how well the model is identifying nodules.
So, we're looking at recall now, instead of F1?
Exactly. High recall is key here. If we miss potential nodules, we're in trouble.
Right, we can't afford false negatives. What about false positives?
The classification model will filter those later. Our current focus is on finding as many nodules as possible. Recall is paramount in this early stage.
The code calculates percentages, right?
Yeah, it computes percentages of true positives, false negatives, and false positives, compared to all labeled candidate nodules.
So, this is per epoch, right?
Correct. We look at these metrics for each training epoch to track improvement over time.
Why do we validate only on certain epochs?
We're only validating on specific epochs, like the first and every fifth, to save computation.
Got it. So we save the best model we see, based on recall. How exactly does that work?
We keep track of the best recall score seen so far. When we save a model, we flag it as the best if the current score is the highest.
So when do we save the model?
Before saving, we check if the current score is the best seen so far. This is done right before saving the images. The code ensures we don't waste resources storing all results for all epochs.
So the best underscore score is crucial in this training loop.
Absolutely. It allows us to keep the best performing model, given our focus on recall.
This is all about optimizing our nodule detection, not just overall accuracy.
Exactly. We prioritize recall in our model selection process, which is an important consideration given the context of our application.
So, saving a model, not the whole thing, just the parameters? Why is that better?
Well, Alfred, saving just the parameters gives us more flexibility. Imagine training a model, and you want to reuse some of its weights in a different model. Saving the entire model ties you to the exact class structure.
Right. So, you're saying it's like a library?
Exactly. You can take those learned parameters and plug them into a different, similar model, even if the exact class isn't the same. It's like using components from a toolbox. You don't need the whole toolbox to use the screwdriver.
Gotcha. And how do we get those parameters?
We use the model state_dict function. It essentially extracts all the learned weights and biases into a dictionary.
Okay. And the script shows this function being used within a bigger function called saveModel. What's the purpose of all that extra data being saved? sys.argv timestamps.
See, Alfred, those are crucial for reproducibility and debugging. Think of syst.argv, all the command line arguments used to run the training. If you want to recreate the training later, you need that information. The timestamp helps you pin down the exact time of the training and pinpoint when different model versions were generated.
So, you're saying you're essentially creating a detailed history of the model?
Yeah, including the optimizer state, and the epoch number and total training samples. It's a complete record, allowing us to resume training precisely where we left off. And the SHA1 hash? That's a checksum.
A checksum? Why is that important?
It helps identify any accidental file corruption or changes. If a file is renamed or tampered with, the checksum will change, and you'll know there's a problem.
Fascinating. So, a backup of the best model is also saved. Why is this important?
Yeah, you only need the version with the best performance. The other epochs are historical data. This way, you don't get bogged down in the minutiae of all the training runs.
You make it sound like there are a lot of potential scenarios in which these methods of saving a model become crucial. That's super important, especially in complex training situations.
Exactly. Precisely tracking model versions and preventing data corruption are key in large-scale training experiments, ensuring you know exactly what model you're using.
So, Samuel, these training results, they're showing some interesting patterns. What are the key takeaways?
Yeah, the F1 score is trending up, which is good. True positives are increasing too, but we're seeing huge false positives.
Huge? Like what for 1000%?
Precisely. And that's expected, given our training and validation data. We're training on smaller crops, validating on full slices.
So, why the huge difference? Can you explain that?
Think of it like this. Imagine a picture of a dog. Training on a small section might identify the dog's ears as a dog. But validating on the whole image could find lots of other things that look like ears in other places, false positives.
Right, so the scale is the problem. But why is the recall plateauing after a while? That sounds concerning.
That's a sign of overfitting. The model is memorizing the training data, not learning the underlying patterns.
So, we're kind of getting our training results, but the validation isn't quite following suit?
Exactly. We see a stark difference, like a disconnect. Training keeps improving, but validation results show this overfitting. It's really important to keep an eye on that disconnect.
And the recall being a priority because of downstream classification models?
Yes, recall is crucial. We can accept some precision trade-offs. Remember, precision is about how often our prediction is actually correct when we say it is. Recall is about how many relevant things we're able to catch in our dataset.
So, we can just rely on the recall to make our choice?
Well, not entirely. We need to be careful. While recall is important, we can't ignore precision entirely. Those false positives might be problematic.
Right. And these validation results, how reliable are they since we have such different scale issues?
We're somewhat cheating, as we know the outcomes of Chapter 14. But in reality, running these experiments for validating data is a must. Educated guesses are great, but direct, real experimentation will get us the results we need. Our current metrics are sufficient for now.
So, we're going forward with these results, but with a clear understanding of the caveats. That makes sense.
Precisely.
So, Samuel, we've built a new model for pixel-to-pixel segmentation. Tell us about the structure.
Yeah, we've used a UNet architecture. It's really good for these tasks. It's kind of like a mirrored, layered network.
Mirrored? Like a reflection?
Exactly. It has a contracting path that extracts features, and then an expanding path to reconstruct the output. This approach helps capture detailed information while maintaining context.
Makes sense. And the dataset's been modified too?
Right. Smaller image crops for training and fewer validation slices. This is a smart way to optimize training.
Why smaller crops?
Well, training with larger datasets is often computationally intense and not necessarily more effective. Smaller crops are a faster, more efficient way to extract critical information.
Clever. And what about the training loop changes?
We've added logging to TensorBoard. Really useful for tracking training progress. Also, moved data augmentation to a separate GPU-based model. Makes training way more efficient.
So, faster training, better data use, more visualization. This sounds ideal. But what about the results? The false positives are different than expected.
That's correct. But our project requirements still allow for these results. It's all about balancing efficiency with accuracy.
Interesting. So, while not perfect, the trade-offs make it acceptable. It sounds like, Samuel, you are making your way towards that cohesive, end-to-end model in the next chapter?
Exactly. Pulling all these parts together for the final project.